that whole background, but it would provide a lot of, future, like for dentists at least, they'll get to know, how much water should they actually apply to the tooth when, cleaning, and if a person, like the age of the person, because different ages, different sensitivity of the tooth, the weaker the tooth is, applying so much water during cleaning might harm the tooth. Or even with like a cavity, cavities make teeth more sensitive, so having this cavity could impact the tooth even more with greater water being applied to it.
Sure. And basically, as I mentioned, even though the research is rudimentary and exploratory, it can provide significant insights, and then best practice can be developed. But again, keep in mind, clinical trials are needed, FDA has to put a stamp on it. And FDA, if the FDA doesn't put a stamp on a device, medical device, you got nothing. But the thing is that you start a concept, hopefully, somebody will pick up on it. Maybe Ms. Rana will further this in her career. That's the hope. But again, collaboration with the dentist is needed, to understand, to run some field trials. I know the computer can produce enough results, very nice graphs, very good data, but all fails if it cannot be proven on the field. So field test is needed. Maybe collaboration with a couple of master's and PhD students in collaboration with the engineering department is needed. So there's a lot of work is needed. Hopefully, I have a lot of hope that somebody will take this research, or maybe Ms. Rana will take this research and further this. Basically, that's what the hope is. The impact of the water jet on other tooth and casting effects need to be captured. As I mentioned, a couple of teeth need to be modeled together, and then the jaw probably the next step, all right, so that the impact of one tooth over the other is needed. Because keep in mind, it's like when you release a jet in your tooth, one is hitting one angle, is hitting the other one. So the coefficient of restitutions and things like that need to be captured. And it's just the whole world is open to you, in fact. And this couple of references we use. Obviously, there's not much of references because we really couldn't find a lot. Out there. I just wanted to run one of the simulations that we have here. So so I'm just running the impact of the water jet here. And if you look right here, there's a pressure. The pressure at the bottom is, so the velocity is higher, the pressure is low. And basically, the velocity starting here is lower than this. So obviously, the pressure, the velocity impacting the gums, all right, is higher than the top over there. And that's the main simulation for that. And I just wanted to show this to you guys that, we kind of model this in the SolidWorks CFD package. And then, again, much more work is needed in this area. Basically, again, increase the number of mesh size and things for that, and at different angles. So that's just wanted to show you. A lot of people like graphics and colors and everything. But again, as I mentioned, these results have to be validated on the field. As an engineer, you want to do that. You want to be absolutely sure that what you're getting on the computer is what you get on the field. That might be on and off a little bit, but again, engineering is an applied area. It's physics, chemistry, mathematics, all together, the knowledge together and applied. And we are very close. If we make sure the bridge doesn't fail at certain stress, it won't fail at certain stress. But again, you got other uncertainties. But we are very close. It's an approximate guesstimate, as I said. All right, but it's a science that backs it up. All right. Well, I'd really like to thank the audience here. Thank Ms. Salam. She has done a fabulous job. Again, as I mentioned, I can't really beat the drum enough. It's a very, very hard subject to understand and synthesize for an undergraduate student. And for her to take this and do such a good job is really commendable. Thank you. Yes, sir.
For the recording, sir. For the recording, you're going to need that.
Thank you so much. I apologize for coming late. But I was teaching and I just finished teaching. But I was just curious because it looks like it's a very nice, interesting application. And so, have you tried to contact dentists in the area and have they showed interest or not? Because I know a couple of dentists that I'm sure might be very much interested. So because you said it needs the cooperation, of dentists and so on. So have you tried to make an effort to contact dentists, or you already have dentists on board that are working with you and trying to see?
That's a very good question, and we wanted to do some kind of work before we approve the dentist, because we want to show that we have done some field work before we go there. So this is the field work, and you're right, sir, that the next step is going to the dentist, because ultimately we can only get valuable insights from that. We can think from our way, but if you don't have a dentist on board with the collaboration, nothing is going to get approved. You're right. Thank you so much. Yeah, that's a wonderful question. Yes, sir.
Thank you for the great work. I'm not an engineer at all, but I would imagine that the model itself, the way it's attached and the way it's fixed, would affect your data about the pressure and the velocity.
Exactly.
So the model, is that fixed in a way that is similar to how the tooth is fixed into that organic soft tissue of the gum and so on?
Absolutely. At this point of time, the tooth is actually fixed in space. So keep in mind, when you look at a tooth, it's like a cantilever beam, like a beam that's hanging out, but it's vertical. All right? So it's fixed in space and time right now. All right? And the fluid is actually flowing over it. The way the software is analyzed is fixed in space. All right? But if you're looking at a soft support, we haven't modeled the soft support. That's the next step, where you take the fluiding forces, and that's the beauty about a CFD. It's called multi-physics software. There's the next level which comes out. We have the software here. There's multi-physics. So you can take or import the fluidic forces and transfer them over to the solid mechanics package or the simulation package, where you can analyze the forces and see the deflection of the tooth and look at the reactions and the support at the gums. That would be a very, very, very valuable piece of information. Yes, sir.
Question is, sometimes damage, when it's acute in the tissues, may not be as harmful, because we do damage sometimes intentionally to improve, because regeneration and so on, this is a living tissue. So I'm just trying to raise these questions because the recycling that happens in the organic tissue and the living tissue makes a correction of damage maybe sometime a positive change. So I know that it's too early to even think about it. It's a great innovation here, innovative work, but these are areas that was drawing my attention when you were talking.
That's correct. And as you say, as we talk about nature takes its course, right? That's what you're hinting at, that nature takes its course, basically. Tissues die, they come back again, right? The thing is that here, if we can prevent intentional damage to avoid long-term implications. The thing is that if you look at, I'll try to use my analogy. I know you're a doctor, you're in the medical area, I'm trying to use my analogy, like a bridge, for example, right? If you look at a bridge, if the air blows over it, it sways back and forth, right? Back and forth, a lot. The design, when the engineers design the bridge, is called fatigue. When the engineers design the bridge, they build that design into it. They know that if the bridge sways widely, or shears, or twists, then have a ripple effect called resonance and everything. They build that design into it. All right? But is that condition desirable? Of course not. You're right. And this is something here we'll try to, and as the research grows, and it becomes more mature, which I hope, maybe this aspects will get looked into. But that's a wonderful question, yeah. But let nature take its course, but if you have to intervene, like life expectancy. It used to be 50, 60, but medical intervention has prolonged the life expectancy, right? So if intervention will help for a better dental hygiene, why not? All right? That's probably not the answer the doctors want to hear, because the livelihood depends on that, right? Yes, sir. Good question.
All right. Basically, if I understood correctly, you had a threshold that you measured for the maximum pressure, and I think you had the value around 600 kilopascals or something like that. And that was for the tooth or for the soft tissues?
That was for the soft tissue. So basically, you're trying to look at the pressure. So keep in mind, If you look at the Newton's philosophy or Newton's law, every action is opposite reaction.
Yeah.
If you're having two 620 kilopascals and it's hitting you this way, the soft tissue is it's going to react to the soft tissue at 620 kilopascals.
Right. My question is actually, is there another threshold on the minimum side that you can, because it seems to me you're trying to generally have effective teeth cleaning with this method without damaging the soft tissue, and even though you have a set threshold, you said that older people might have lower thresholds and so on and so forth. So is there any literature about a minimum threshold of pressure that will have effective teeth cleaning capabilities?
And that's the right question, a good question. I'll tell you why didn't we shoot for the minimum? Because the industry is striving. See, the thing is that industry is striving for higher velocities. Why? Because higher velocities one day, they expect that, if you go to a dentist, they try to scrap your tooth and try to remove the gingivitis and the plaque buildup and everything using tools, which is very uncomfortable, right? They hope that with water jet, with very high pressure, high velocity, they can probably replace that. You see that with multiple or maybe more frequent visits, or you can do it at home, right? But so the more the velocity, the merrier it is. So we didn't try to shoot for minimal what is there, because there's always a strive, there's always a pursuit for higher velocities.
Thank you.
Yes. Any interesting questions?
Any other questions? Thank you, Dr. Agrawala. Thank you, Reina, for this wonderful presentation. Thank you. We appreciate your
Thank
time.
you very much. I appreciate it.
Thank you. Okay, our next presenter is Dr. Ahmed Al-Hasni. He's presenting a paper on FTR ABCD, is a low iron and acid-induced ferrous iron specific transporter that is required for the virulence of Brucella abortus in mice. Dr. Ahmed Al-Hasni is a PhD candidate in the Department of Microbiology and Immunology, Brody School of Medicine, East Carolina University. He has a master's degree in microbiology from Zagazig University in Egypt. Currently, he is doing his PhD in Dr. Roop Lab, which studies metal acquisition systems in Brucella and how they affect the virulence of these bacteria. Welcome, Dr. Al-Hasni.
First, I'd like to thank Dr. Nihad for giving me the opportunity to be here, and I'd like to thank you guys for coming. Hopefully you'll find my topic interesting. So I'm going to talk about FTR ABCD, which is a low iron acid-induced ferrous iron specific transporter that we have recently discovered in Brucella abortus, and we have found that it is required for the virulence of these bacteria. I will give a short background about Brucella. Brucella are important zoonotic pathogen. They are facultative intercellular gram-negative bacteria. They are not free-living microorganisms. They live exclusively associated with their mammalian hosts. Brucella can infect many animal species. They prefer to colonize the reproductive system, causing infertility in male and abortion in female. Human can acquire infection via contact with infected animals or consumption of their products, especially milk and milk products. And infection in human is called human brucellosis or andurate fever or Malta fever. It's a very bad disease. It's like having flu for a long time. It requires treatment with combination of 2 antibiotics for a long time, for maybe like 2 months. And sometimes there is a relapse. And currently there is no safe vaccine for human. There is vaccine for animals, but there is no safe vaccine for human. And brucellosis is the most common bacterial zoonotic disease worldwide, with more than 500,000 cases every year. In the US and other developed countries, the disease is controlled due to the what's called the eradication and effective vaccination programs that had started like 30 years ago, but the disease is still endemic in many parts of the world. And worldwide economic losses due to brucellosis are huge in terms of decreased animal production and the cost of treatment for human and decreased their productivity. So the most pathogenic species for human are Brucella abortus. This is the microorganism that we work with. It mainly infects cattle. The second one is Brucella melitensis, which infect goat and sheep. And the third one is Brucella suis, which mainly infect pigs. As I said, human can acquire infection via contact with these animals or consumption of their products. The problem with these bacteria is that they can easily aerosolize, and they are highly infectious via the aerosol route. So that people who, for example, work with Brucella in the lab like us can become infected by like exposure to these bacteria. This is why when we work with these bacteria, we work in special labs called BSL3 lab, Biosafety Level 3 labs, to protect ourselves. Now, Brucella, as I said, they are intracellular microorganisms. They live mainly inside macrophages. And the ability to cause chronic infection, which is the hallmark of human brucellosis, is due to their ability to survive within macrophages for a long time. And macrophages are perfect environment for these bacteria. I will talk about this in a minute. So after being engulfed by macrophages, they live inside vacuoles called Brucella-containing vacuoles. These vacuoles are acidic in nature in the early stage of infection due to interaction with the endolysosomal pathway. So for this bacteria to avoid fusion with the lysosome and killing by the enzymes and bactericidal compounds in the lysosome, they start to change their trafficking pattern inside macrophages. So this acidic pH stimulates what's called type 4 secretion system. It secretes effector molecules, which change the trafficking pattern inside macrophages. So they start to interact less with the endolysosomal pathway and interact more with endoplasmic reticulum. As a result of this interaction, the pH inside these vacuoles rise. And by 24-hour post-infection, these vacuoles fuse with the ER, forming what's called replicated Brucella-containing vacuole, within which Brucella can replicate. Now, our lab is interested in studying metal acquisition system in Brucella, especially iron. Now, as you know, iron is essential micronutrient for nearly all organisms. It's required for many biological processes like oxygen transport and storage, nitrogen fixation, photosynthesis, DNA biosynthesis, citric acid cycle. Because it's enter in the structure or required for the activity of many enzymes involved in these biological processes. Iron exists mainly in two redox states. The main redox state is the ferric form, which iron 3. The problem with this ferric iron is that it is poorly soluble at normal pH, at neutral pH. This leads to the problem of poor bioavailability. The second form is the ferrous iron, which is iron 2. This form is relatively soluble at neutral pH. However, it is, can easily oxidize it to the ferric form when it is exposed to air at neutral pH. This is why it is restricted to anaerobic or low pH environments. And this graph shows that this ferrous iron is very sensitive to oxidation at pH 7 when it's exposed to air, while it is resistant to oxidation at pH 4. I'll talk about why this is important when I explain my results. Now, Brucella, like other microorganisms, require iron. However, acquiring enough iron is a big challenge for these bacteria because they live inside macrophages, and So, as a part of innate immune response to the infection by Brucella, interferon gamma causes activation of macrophages. And this activation causes depletion of intracellular iron levels via 3 mechanisms. So, when interferon gamma activates macrophages, it decreases the transferrin receptor. These receptors are responsible for acquiring ferric iron from the circulation in the form of ferric transferrin complex. So it decreases the uptake of ferric iron from the circulation. And also interferon gamma stimulates the ferroportin exporter, which exports ferrous iron from inside the macrophages to outside. So it it decreases the intracellular iron level. And the third mechanism is stimulation of what's called NRAMP1 transporter, which exports divalent cations, including iron, from inside this vacuole to the cytoplasm. So overall, the intracellular iron level inside macrophages will be very low to support the survival of these bacteria or any intracellular pathogen. So bacteria, to overcome this, like iron withdrawal strategies, they have iron acquisition system. It's like a battle or a challenge between bacteria and the hosts. Brucella has like 5 or 6 iron acquisition system. It has heme acquisition system. It can acquire heme. So, as I said, macrophages are perfect environment for Brucella because macrophages play an important role in the recycling of senescent RBCs. So these senescent RBCs become engulfed by macrophages, and then the gluten part become degraded, and then which releases heme. Brucella has a system that can acquire this heme and use it as an iron source. And also it has 2 CD4 dependent uptake system. CD4, it's like iron chelator, which like bind iron with high affinity, and then become transported into inside the cell. And also they have free iron uptake systems. And recently we have discovered that it has ferrous iron specific transporter, which we call FTR-ABCD. Now, these 4 genes, which we have given the name FTR-A, B, C, and D, we have found that they include components of what's called FTR1B based ferrous iron transporter, which is homologous to ferrous iron transporter that have been recently characterized in other bacteria like Bordetella and E. coli. This is a new type of ferrous iron transporter. The activity of this new type of bacterial ferrous iron transporter depends on homologue of eukaryotic FTR1B permease. So these bacterial ferrous iron transporter, for their activity, they require a permease which transport iron across the cytoplasmic membrane. This permease is homologous to the eukaryotic permease called FTR1B. FTR1B is a part of high affinity ferrous iron transporter that is well characterized and studied in Saccharomyces and other lower eukaryotes. This is a model of how this ferrous iron transporter works in Brucella and Bordetella. It consists of 4 components. FTR-A, it's a periplasmic ferrous iron binding protein. So we expect that this protein binds ferrous iron that diffuses across the outer membrane into the periplasm, and then it binds FTR-A. FTR-B, we think it is an oxidase, which oxidizes ferrous into ferric. And then the produced ferric is transported across the cytoplasmic membrane through FTR-C, which is high affinity permease. It's homologous to the Saccharomyces FTR1B. And then the released electrons from oxidation is transported across FTR-D, which is integral membrane protein, we call it polyphoredoxin. It has iron-sulfur binding domain. So basically what this protein does is that accept electron and transport them into unknown oxidant. So it's some, you can think it's complicated. Why like ferrous is not transported directly? So so we think like this system is more efficient in ferrous ion transport than other ferrous ion transporter, because when you oxidize ferrous into ferric, so you shifting the equilibrium so that more ferrous will get into the cell. So other ferrous transporter like can transport ferrous like directly, but when you like oxidize ferrous into ferric, there will be like less ferrous inside the cell, so the equilibrium shifts inside, making more iron go inside the cell. And why also like this oxidation step, they think it's important to protect this protein from the damaging effect of ferrous iron. So iron has a damaging effect, as I will show in a minute. So this is why we think this oxidation step is important. So to study this transporter, we deleted the first gene of this operon that encode the periplasmic binding protein, iron binding protein. And then we measured the radiolabeled iron uptake. So we compared the ferric and the ferrous, or the radiolabeled ferric and ferrous iron uptake with the wild type. And we found that our mutant has a significant defect in the uptake of radiolabeled ferrous iron compared to the wild type. So this is our mutant, and this is the wild type and the reconstructed strain. Reconstructed strain is the strain that we put back our gene. So our mutant has significant defect in ferrous iron uptake, but it has no defect in radiolabeled ferric iron uptake. So this confirms that our transporter is ferrous iron specific. And then we wanted to test if this transporter is required for variance. So the ultimate goal of our work, we are looking for variance factor in Brucella. Because like Brucella, there's no safe vaccine for human. So if we find like a variance protein or a protein that's required for virulence, like someone else may use this protein as a candidate for making a vaccine, because now there is no safe vaccine for human. So we tested if it is required for virulence. So the way we test virulence in Brucella, we use either macrophages or we use macrophages and mice. So we test the survival inside macrophages. So to do this, we take macrophages from the peritoneal macrophages from mice, and then we put these macrophages in 96 well plates, leave them like overnight in DMM media, and then we infect these macrophages with opsonized Brucella. And then after certain time points, 2, 24, 48 hours, we lyse these macrophages and plate the Brucella on blood agar to count how many intracellular Brucella are there or could survive inside macrophages. Using this, we have found that our mutant, this is our mutant, it has significant defect in survival inside macrophages after 24 and 48 hour post-infection compared to the wild type and the reconstructed strain. So we were excited about this because we were the first to show that this type of bacterial ferrous ion transporter is linked to virulence. So this ferrous ion transporter in eukaryote have been linked to virulence, but in bacteria no one has shown before that it is required for virulence of bacteria. And also we tested the virulence in mice. So the way we do this, we inject mice intraperitoneally with Brucella, and after 1, 3, and 5 weeks, we sacrifice the mice, and we take the spleen out, homogenize it, and then take the homogenated sterile diluted and plate it on blood agar, and then we count how many Brucella were able to colonize the spleen of the mice. And we found that our mutant is significantly attenuated in mice after 3 and 5 weeks post-infection. So this is our mutant. It's not attenuated after 1 week, but it's attenuated after 3 and 5 weeks. This is because like the immune response is not fully developed or doesn't have like the full strength until like 2 or 3 weeks post-infection. And as I told you before, interferon gamma activation it is the main factor which causes the depletion of intracellular iron level. Now, the identification of ferrous iron transporter in Brucella, we think it is important, or we believe it is important, because as I said, Brucella in the early stage of infection live inside vacuoles which are acidic in nature. So this acidic pH, as I showed you before, favor the stability and solubility of ferrous iron. So ferrous iron is a relevant iron source for Brucella. Now, I'll talk about the regulation of this transporter. Did I exceed my time limit? Or? Okay, I'm sorry. I'll talk very fast. So, too much iron is toxic for the cells. So this is why bacteria, including Brucella, has to regulate their iron oxygen system. Iron, especially ferrous iron, can react with reactive oxygen species like hydrogen peroxide, producing highly reactive hydroxide radical in a reaction called Fenton reaction. This hydroxide radical is very reactive, can cause damage for the DNA, lipid, and protein. This is why bacteria, including Brucella, has to regulate their iron oxygen system. So we have in Brucella, we have a protein called IRR. This is the main iron responsive regulator. So this regulator acts as, like under low iron condition, it acts as an activator for iron oxygen system, like our transporter. At the same time, it represses the expression of proteins which consume iron. And this makes sense too. So you have low iron, so you need to express iron oxygen system, increase the expression of iron oxygen system, and suppress the expression of protein which consume iron. We have found that the expression of our transporter is responsive to low iron condition. This makes sense because it is an iron oxygen system. So using this is real-time PCR, we have found that this is our wild type. 238 is our wild type. Using real-time PCR, we found that the expression is like 15-fold induced in response to low iron, and this iron responsive expression was like abolished in our IRR mutant and was restored in the complementary strain. So IRR is responsible for the low iron expression, responsive expression. Also, we found that the expression of our transporter is responsive to low pH. So when we compare the expression at in iron-rich media, so there's no low iron is not a factor right now. Compare the expression at pH 5 and pH 7, we found that the expression at pH 5 was induced like 25-fold, again using real-time PCR. And this like acid responsive expression, we think it is like important because, as I said, this transporter, we think it's important in the early stage of infection where Brucella live inside this acidic vacuole. So this acidic pH can stimulate ferrous iron uptake. Because under this acidic pH, the predominant iron form, we think it's ferrous. Last thing, we wanted to know if our regulator IRR has a role in this acid responsive expression. And it turned out that no. So we have two environmental stimuli that affect the expression of our transporter, the low iron and acidic pH, and they are our transporter is independently regulated by these environmental stimuli. So using this beta-galactosidase assay, we found that expression is responsive to low iron and low pH. Maximum expression was achieved at low iron and low pH. And IRR doesn't have like a role in the expression in response to low pH. Because in IRR mutant, the expression, you see this one, in IRR mutant, the expression was still responsive to low pH. This is the wild type and this is the mutant. The expression is less than the wild type, but still responsive to low pH. So there is another regulator which is responsible for the expression of our transporter response to low pH, other than IRR. And we're currently working to identify this regulator. And I skipped the summary, and I'd like to thank my mentor, Dr. Rube, and my committee member. and I'd like to thank you.
Any questions?
Thank you very much, Dr. Ahmed, for this great presentation. Actually, I have a bunch of questions after this presentation, but let me summarize them in one line. The first, I'd like to ask, how many strains did you use before injecting the macrophage with the Brucella? So have you used all the strains of the Brucella, or it's only one strain that
We're
you
working
used?
with one strain.
The second thing, you mentioned 2 experimental studies that you did, one with the macrophage after extracting them from the mice, but before being activated with the interferon gamma. And the second experiment, you studied the spleen growing up in the mice after injecting with the Brucella. So, but you didn't, you didn't test the effect of the macrophages inside the mice, because this will gonna change the response after being activated with the interferon gamma inside the mice. So you are testing the macrophage in vitro, and this doesn't implicate the same thing in vivo.
So for the experiment, it can be done with activated macrophages. So sometimes we can add interferon gamma in vitro.
So you did this in that study?
No, I didn't do this because sometimes when we add interferon gamma, our mutant dies. Even the wild type can die. It cannot survive. So sometimes, or most of the time, we do it without interferon gamma activation. It doesn't mimic the actual environment, but it gives us an indication. So we don't. This is why we don't just do the macrophage experiment. We couple this with the mice experiment.
Okay, Dr. Ahmed, this is a great presentation. In liver disease, there is a disease called hemochromatosis, and that's a problem with the iron. And in the enterocytes, there is a protein called hepcidin. The hepcidin, what it does, it inhibits the ferroporotin, and it keeps the level of iron inside the enterocyte high. Do the macrophage have hepcidin too?
Yes, I think so, but
did you
did
you come across hepcidin in regulation for iron metabolism inside the macrophage, or that's something probably more specific to the enterocyte?
So there is a response called hypoferremic response. So, but this is a response to extracellular infection. So as a result of extracellular infection, as I remember, hepcidin can cause sequestration of iron inside the cell, so that it limits the iron for the extracellular bacteria. Yes. Yes. Yeah. So it inside. Keep it inside. But this iron overload. Exactly. So this affects the extracellular bacteria, but not the intercellular. Brucella are intracellular bacteria.
Great work. I think it can lead to great results. How do you think this work can lead to some methods of eradication of brucellosis?
Okay, so there are some studies that have shown that targeting iron acquisition system can be used as antimicrobial agents. So by targeting iron acquisition system, you can kill the bacteria. But this is not for sure. But some sites suggest this. Our ultimate goal is finding, or our results suggest that we have a very attenuated mutant. The FTRA mutant is attenuated in mice and macrophages. So this can be used as a candidate for live attenuated vaccine. Not us. We are just doing the basic research. Someone else may use it as a candidate.
Do you think any antibody to FTR receptor transporter may result in decreasing the virulence of brucellosis?
So it's a periplasmic protein, so it's not like expressed. This is why we don't think there will be antibody for it.
Any other questions? Thank you, Dr. Al-Hasni. It's a great presentation. It's a great work. Thank you. Okay, our last but certainly not least presentation. Our last presenter is Mr. Karim Kandil. He will be presenting a paper called Effect of 6-Week Weight Loss Program Prior to PEG-IFN, Ribavirin Therapy of Chronic Hepatitis C on Early Virological Response. He has a team of other scholars, Dr. Sharif Al-Bahiri, Dr. Hani Al-Wakil, Dr. Laura Mataris, Dr. Islam Ali, Dr. Glenn Harvin, Dr. Saeed, and Dr. Hussam Kandil from the Division of Gastroenterology, Hepatology, and Nutrition Department at East Carolina University, as well as the Departments of Statistics at East Carolina University. Please welcome Mr. Kandil. Mr. Kandil received a dual degree in bioengineering and psychology in 2013 from the University of Pittsburgh. During his undergraduate career, he published 3 papers with the University of Pittsburgh Medical Center's Western Psychiatric Institute and Clinic while working in their genetics lab, and is now currently working for East Carolina University as a research assistant. Welcome, Mr. Kandil.
All right, thank you so much for that double introduction. All right, so as was already said, I'm going to talk about the effect of a 6-week weight loss program on pegylated interferon and ribavirin therapy of chronic hepatitis C on the early virological response. So, chronic hepatitis C is currently a significant global health problem. 3% of the world's population is currently infected with hepatitis C and is responsible for up to 20,000 deaths a year. 20% of people with hepatitis C will develop cirrhosis and hepatocellular carcinoma, and it's the most common cause of liver transplant in the United States. So you can see we have a map here of the prevalence of hepatitis C worldwide, with the darker areas being the areas with more prevalence of hepatitis C. As I said, 3% of the world's population, more than 170 million people worldwide have hepatitis C, including 4 million Americans. Of those 170 million people, 20% worldwide are classified as obese. So, hepatitis C treatment has been progressing rapidly over the last several years. There was regular interferon that was given subcutaneously 3 times a week with ribavirin, which had a roughly 20% response rate amongst patients. The treatment we used, the pegylated interferon, which was only done once a week instead of 3 times weekly, with ribavirin as well, and that improved the response somewhat. And in more recent years, in the last couple of years, there have been direct-acting agents that have been added to the therapy, which have further increased response rate, now bringing it up close to 80% for most patients. Several factors affect the response to treatment from patients. The genotype of the virus, with genotypes 1 and 4 having the lowest response rate, roughly 40%, with genotypes 2 and 3 having almost double the response rate of those genotypes. The extent of fibrosis in the liver can affect the response to treatment. Race, with African Americans and Hispanics having much lower response rates to treatment than Caucasians and Asians. And hepatic steatosis in the liver as well can affect. What we want to focus on is obesity, which has been recorded to affect response rate by 50%. Several mechanisms have been proposed for why obesity affects the response rate to the viral treatment so adversely. Firstly, we have decreased interferon bioavailability. As there's a larger weight distribution amongst obese patients, less of the antiviral treatment can reach the necessary areas, and it's also harder to be absorbed through fatty tissues because of reduced lymphatic drainage. Lamb, in his publication, showed that obese patients with chronic hepatitis C had lower concentrations of interferon in their systems than non-obese patients when given the same dose of interferon. So also, obese patients tend to have more fatty liver than non-obese patients, and there are several mechanisms that have been proposed for why fatty liver may cause a decreased response to treatment. So whenever fat is deposited in the liver, there's a decreased contact area between the drug and the hepatic membrane. It alters the liver function because of the higher concentration of fat in the tissue, and it distorts the normal architecture of the liver, and all of those may lead to diminished hepatic response to the interferon. Patients who have chronic hepatitis C and are obese are more likely to also be insulin resistant and have more advanced hepatic steatosis and steatohepatitis and fibrosis than their non-obese counterparts. And obese patients are more likely, therefore, to be non-responders to combination therapy and are relapsers when compared to the general population. So our study is designed to determine the impact of weight management before starting the hepatitis C therapy on treatment response amongst the overweight patients. Specifically, we want to study the effect of a 6-week weight management program prior to starting the PEG interferon and ribavirin therapy on early virological response. We want to compare this early virological response for overweight subjects who experienced significant weight loss versus those who did not experience such weight loss. For the purposes of our study, we defined significant weight loss as more than 3% of their initial body weight. So the study is an interventional, prospective, randomized, case-control clinical trial. Early virological response was defined as a 2-log drop of the viral load, and early virological response has been shown to be a strong predictor of sustained viral clearance, or more or less that they're cured. As I said earlier, 3% weight loss was chosen as a cutoff, and this was because studies have shown that this is the cutoff for significant this is the point at which there's decreased steatosis and decreased inflammation amongst hepatitis C patients. So our inclusion criteria for patients in the study is that they're above 18 years of age, that they have hepatitis C of the genotype 1, and we confirm that by PCR, that they have no other liver disease, negative for HIV, no cardiac, renal, or liver failure, or cancer, and no contraindication to chronic hepatitis C therapy. So 64 patients total met our inclusion criteria and were approached by our team. 36 agreed to participate in the study. Firstly, Group A, which is our lean control, all patients with less than 25 BMI, had 11 subjects. Then the rest, the overweight or obese patients, were randomly divided into one of two groups. First, Group B, which is our standard of care control, who received a brochure and 5-minute discussion about weight management with a dietitian, just our minimal standard of care for obese patients. And Group C, who received a more rigorous 6-week weight management program. The viral load of these patients was checked by polymerase chain reaction at baseline and at 3 months, at the end of treatment after 12 months, and then 6 months after that after the completion of therapy. The weight and the height were also recorded at initial visit, start of therapy, and 12-week point of therapy. For all of these groups, standard PEG interferon and ribavirin treatment was started 6 weeks after the initial clinical visit. During those 6 weeks, Group C received their weight management program, and so all of them received treatment at the same time. This weight management program consisted of a 6-week educational session on nutrition. The patients were encouraged to start with 10 minutes of physical activity and gradually increase up to 30 minutes each day. The dietitian provided each subject with a diary for daily recording of their dietary intake, and every patient was given a pedometer to record their daily physical activity. All right, so then these Group C patients were given monthly follow-up sessions to review their diaries, their pedometers, and their patient records of physical activity, discuss any diet-related issues that they ran across, and reinforce and encourage compliance to dietary and physical exercise recommendations. All right, so on to our results. Here we have the demographic data for all of our patients. The only significant difference between the three groups was that Group A was significantly younger than the B and C groups, though this can be expected with obesity being higher in older age groups. And for the rest of the demographic data in terms of race, gender, and so on and so forth, there was no significant difference between the groups, at least statistically. All right. So here we have the weight changes at the start of hepatitis C therapy and at 12 weeks of hepatitis C therapy. And on the left, see if we can move it over here. On the left, you can see that these three show all three groups weight changes at 0 weeks. So this is after the 6 weeks that the Group C went through the intensive weight management program. And there wasn't any significant, though the Group C did lose some weight, there wasn't any significant difference between the three groups in terms of weight loss. And after 12 weeks, all three groups lost weight, with C tending to lose more, though again, this change was not found to be statistically significant. Here again, we have the same data, but with percent body weight instead of kilograms. And again, same results. The change in weight was not found to be statistically significant at the start of therapy or at 12 weeks. Here's the early virological response for all three groups, with 80% of Group A showing an early virological response compared to roughly 50 in Groups B and C. There's no statistically significant difference between the B and C groups in that regard. And here we have the effect of at least 3% weight loss on EVR amongst the overweight subjects. So here we combine the B and C groups, because those are all of our overweight and obese patients, and wanted to determine whether weight loss experienced by those would affect their response to treatment. So you can see that when on the left here we have all of the patients who did not see significant weight loss. So they had no weight loss or weight loss under 3% of their initial body weight. And on the right, the patients who did have at least 3% loss of body weight. So you can see that the ones who did have significant weight loss lost significantly more, had significantly more response to treatment than their non-weight loss counterparts. So in conclusion, overweight chronic hepatitis C patients on a dietary and exercise program for 6 weeks prior to hepatitis C therapy did not lose significantly more weight at 12 weeks than those who just had a one-time discussion and weight management brochure from a dietitian. Overweight hepatitis C patients who lost more than 3% of body weight at 12 weeks had higher EVR compared to those without 3% weight loss. And weight loss prior to hepatitis C treatment may improve treatment response in overweight patients. All right, thank you for your time. I now open up the floor to questions. You gonna help?
Any questions to Kareem? Questions? I actually have a question, and again, I just want to look back at the results because I can't remember at the top of my head.
Which one?
So, yeah, the one basically where you combined the two. So the green on the right hand side, these are the combined.
Yeah.
The groups B and C, the obese people, the extremely obese people.
Yes.
So basically, what were the differences between the three groups initially? Was it the fact that they were not, they didn't like I thought that the A group was lean.
Yeah.
They were lean, right?
Yes. That's why this data does not include the A group. The thing is that
so the result excludes the A group.
Right. Again, the purpose of the study is to see what weight loss, how that affects response to treatment. And so the separation of the B and C group was intended to force a difference in weight change between the three groups. And even though there was some difference, it wasn't statistically significant. So instead, because again, there are going to be some patients who do and some patients who do not lose weight amongst both groups, we just combined that and then just looked at the differences between those two groups instead, the group that did and the group that did not lose weight. In further research,
are you using this to promote you're using this research to promote people to actively, watch their weight, talk to professionals when they indeed have a problem, or like again, because we know obesity is a problem for for a lot of people, and are you trying to increase, how are you trying to increase basically people's awareness to this?
Well, it's not specific. I mean, of course, obesity has far-reaching health problems for many diseases and many disorders. It's been linked to way more things than just hepatitis C. But basically, with our purpose for the study is to illuminate the effect that weight change has on hepatitis C and its treatment only. And we're not really reaching into any more far-reaching implications of obesity.
Or try to look at the study. I mean, I understand this might be maybe a future step in countries that are heavily infected with, hepatitis C. I mean, you've shown us.
That's. I was going to say that in future studies, we'd want to look at maybe larger groups. Recruitment is an issue. So if we go to, again, a country with a higher concentration of hepatitis C, you're more likely to find patients for recruitment. The other thing is that in the future we might want to change our weight management program. Clearly there was not significant weight loss in this study based solely on our 6-week weight management program. So in the future we might implement or alter our current weight management program or implement a completely different one in order to see different results.
This is a great presentation, great results. Thank you. Any other questions? Thank you, Mr. Kareem. Thank you for a wonderful presentation. With this, we conclude our International Scholars Symposium for 2014. Thank you all for participating. Thank you for coming. Thank you for making this event possible. And again, I would encourage you to, always always participate, right? This is not, and I mentioned this at the beginning, this is not an event just for international faculty. This is actually an event that we're trying to foster collaboration between different departments, different divisions, different research, get a flavor of what others are, are talking about and what types of research is going on within ECU. So thank you so much all for coming. Thank you for participating. Thank you for all my presenters today. A great job, great research, potential, and good luck to all of you in the future. Thank you so much.